The Hybriscan system, an in situ hybridization technology for microbial quality control. The Hybriscan system can reduce your time to result by 2 to 14 days, depending on the target organism, while keeping the costs low. It allows the genus and species-specific detection of viable microorganisms only. Sandwich hybridization is not susceptible to matrix interference. The Hybriscan technology can be used for alcoholic beverages such as wine and beer and non-alcoholic beverages like soft drinks and juices. Samples can be prepared in several ways. For liquid samples, after filtration, incubate the filter in the enrichment broth or directly inoculate your sample in the enrichment broth. The Hybriscan system is media independent, so any good enrichment media can be used as the method is not sensitive to the matrix. Examples of typical enrichment media are NBB broth for beer samples and BAT broth to detect and identify alicyclobacillus in fruit juices. It is also possible to pick colonies from plates. Give some glass beads into a 2 milliliter microtube using a spatula. Add 10 microliters of lysis buffer A and 40 microliters of lysis buffer B. Choose and transfer a single colony from an agar plate and resuspend it in the solution. Incubate the mix for 8 minutes on the thermoshaker. The next steps are to open the cell walls and release the ribosomal RNA. After mixing the sample carefully, 50 microliters of lysis buffer C is added, and the tubes are incubated for 8 minutes in the thermoshaker at 37 degrees Celsius and 1400 RPM. Centrifuge at 14,000 RPM equal to 16,000 G for 5 minutes. In the meantime, prepare the appropriate number of wells in the micro-titer plate. With the hybridization solution and pre-warm the plate in the preheated thermoshaker. For hybridization with the probes, 10 microliters of the standards or the supernatant from the sample extract are added to the corresponding wells of the prepared microplate. Cover the microwell plate with a lid and incubate for 15 minutes at 50 degrees and 500 RPM. Discard the supernate and wash the plate with 200 microliters of washing buffer. Incubate for 2 minutes at room temperature, then discard the solution. Add 100 microliters of prepared enzyme solution and incubate for 20 minutes at 25 degrees and 500 RPM on the thermoshaker. After incubation, there are 2 washing steps. For the substrate reaction, all liquid is discarded. Then 100 microliters of substrate solution G from the vial with the green cap is added to each well. Incubate for 2 to 15 minutes at 25 degrees and 500 RPM in the thermoshaker. Positive samples turn blue. This can serve as an early indication of a positive test result. The substrate reaction is stopped with 50 microliters of stop solution H from the vial with the green cap. The color of positive samples changes to yellow. The intensity of the color is proportional to the concentration of target organisms in the sample. If an enrichment step was performed, it is possible to detect, confirm, and identify the organism, but not to quantify it. Place the microplate in a microplate reader and measure the optical density in each well at 450 nanometers. The blank and negative samples should be white to slightly yellow, while positive samples and controls should show a stronger yellow tone. To see our complete portfolio of detectable species, genera, and organism groups, or the detailed Hybriscan operating instructions, visit our website.